Our today's topic is citric acid cycle. Citric acid cycle, which is also known as tricarboxylic acid cycle or the Krebs cycle, after its discoverer Sir Hans Krebs, is the major energy-yielding metabolic pathway in cells and providing the greater part of the reduced coenzymes that will be oxidized by electron transport chain to yield ATP, that is adenosine triphosphate. Citric acid cycle is also the major pathway for interconversion of 4 and 5 carbon compounds in the cell, and many of which arise from or are intermediate in the synthesis of amino acids. Oxaloacetate, which is the cycle's final product, is a key intermediate in the cycle and is also the main precursor of gluconeogenesis in the fasting state. Acetyl-coenzyme a significant carbon input into the citric acid cycle, can be derived from glucose or fatty acids. However, a substantial portion of this acetyl-coenzyme A comes from glucose, more specifically from pyruvate. So pyruvate dehydrogenase complex facilitate the enzymatic conversion of this pyruvate into acetyl-coenzyme A. Now, different steps of citric acid cycle. So first is citrate synthesis. So here enzyme citrate synthase catalyzes the condensation reaction of acetyl coenzyme A and oxaloacetate to form citrate, right? So initiating the citric acid cycle. Second step is isomerization of citrate. Here enzyme aconitase, this enzyme with an iron sulfur center, it facilitates the hydroxyl group migration that makes isocitrate out of citrate. And third step is oxidative decarboxylation of isocitrate. Decarboxylation means removal of carbon dioxide. So first step, this is the first step of citric acid cycle that produces a reduced coenzyme. Here, isocitrate dehydrogenase oxidizes isocitrate and releasing carbon dioxide molecule, right? And reduces NAD plus to NADH, right? And a proton, H plus. So isocitrate dehydrogenase this enzyme is allosterically regulated. Means here ADP and calcium ions activated while ATP and NADH inhibits its activity, right? Next step is oxidative decarboxylation of alpha ketoglutarate by alpha ketoglutarate dehydrogenase complex. Actually, alpha ketoglutarate dehydrogenase complex having 3 enzymes, right? E1, E2 and E3, this complex having 3 enzymes, right? E1 oxoglutarate dehydrogenase, E2 dihydrolipoyl succinyl transferase enzyme, and E3 dihydrolipoyl dehydrogenase, right? It also requires some cofactors like thiamine pyrophosphate, lipoic acid, coenzyme A, FAD, and NAD, etc. Right? So E1 of this alpha ketoglutarate dehydrogenase complex decarboxylates alpha ketoglutarate and transfers 4 remaining carbons to thiamine pyrophosphate, its first cofactor, right? Then E2 transfer the succinyl group to coenzyme ASH, right? This CoASH means coenzyme A synthetase. Here, when it is not attached to an acyl group, right? So with the help of FAD, cofactor. And finally, E3, resynthesize FAD along with NADH from NAD plus so that dehydrogenase complex maintains the substrates and cofactors which are necessary for continued reactions, right? So already, as I told you, cofactors required in this complex are thymine pyrophosphate, lipoic acid, coenzyme A, FAD, and NAD+, right? Next step is cleavage of succinyl coenzyme A. Here, enzyme succinate thiokinase catalyze this cleavage of succinyl coenzyme A, right? Division of this high energy bond, that is thioester bond. Succinyl coenzyme A have a thioester bond. Division of this high energy bond is coupled with phosphorylation of guanosine diphosphate, GDP, and therefore produces GTP in addition to succinate. Right? Next step is oxidation of succinate. Here enzyme succinate dehydrogenase oxidizes succinate to fumarate. Succinate to fumarate. You can see here this reaction. And also producing a reduced FADH2 from FAD. This reaction, right? So next step is hydration of this fumarate. Here enzyme fumarase is the catalyst in hydration of fumarate to maltate. This reaction is reversible, right? Next is oxidation of maltate. Maltate dehydrogenase enzyme here work that it catalyzes maltate's oxidation to oxaloacetate, reducing NAD plus to NADH and producing the final NADH, right? And oxaloacetate here again at the starting point, and this way cycle continues. Now, what are the functions of the citric acid intermediates? Citric acid intermediates may leave the cycle to biosynthesize other compounds. Citrate can be diverted to fatty acid synthesis. Fatty acid synthesis, it includes alpha-ketoglutarate to amino acid synthesis or synthesis of neurotransmitter and purine synthesis. Succinyl coenzyme A to heme synthesis, maltate to gluconeogenesis, and oxaloacetate to amino acid synthesis, right? So intermediates can also be inserted into citric acid cycle to replace catachlorotic process and ensure the cycle continues, right? So this is all about citric acid cycle.